Hi, I'm Camilla.
And I am Oscar. Today we will be sharing everything that we have learned over the past month in our biochemistry unit. We will be breaking down 3 of the 4 major biomolecules and exploring the structure and function of an enzyme.
Let's start with the 4 major biomolecules. There are lipids, nucleic acids, carbohydrates, and proteins. We will be looking at lipids, carbohydrates, and proteins today. Here are a few reactions that make or break the bonds for these biomolecules.
Condensation reactions. A condensation reaction occurs when 2 or more molecules combine to form a larger molecule, releasing a smaller molecule in the process. Smaller molecules are usually water molecules but can also be hydrogen, chloride, or methanol. Hydrolysis reaction. The breakage of a chemical bond by the addition of water or a base that supplies the hydroxyl ion. It is referred to as an hydrolysis reaction. A covalent bond is broken and 2 new bonds are established, each involving either the hydrogen or hydroxyl element of the water molecule.
Proteins are biomolecules made up of amino acids that are encoded by our genes. They are used to repair and build. Amino acids are the monomers for proteins. When 2 amino acids bond, they create a single peptide bond. If a third amino acid joins, a tripeptide bond is created, and any further amino acids that bond to this will create a polypeptide. When a peptide bond is formed, a condensation reaction takes place. This means a water molecule is released when the 2 monomers react. Therefore, to break a peptide bond, a hydrolysis reaction is needed, as a water molecule will separate the 2 monomers. Proteins have 4 levels of complexity. The primary is the unique sequencing of amino acids that bond to create the polypeptide. The secondary is the folding or coiling of the polypeptide. Folding will result in a beta-pleated sheet, and coiling will result in an alpha helix sheet. The tertiary is a 3D structure formed from the beta-pleated or alpha helix sheet. Structure is also supported from the R groups within amino acids. The quaternary is formed from tertiary proteins, and these create the final and functional protein. The sequencing of the 3 DNA bases within the amino acids determine the folding of the 3D structures. The 3D structures determine the function of the protein, as the arrangement of the protein is what allows it to play its role.
Carbohydrates. Carbohydrates are energy-rich biomolecules that are made up of carbon, hydrogen, and oxygen. They are found in sugars, starch, and fiber. Carbohydrates. Aldehydes or ketones that are generally produced carbonyl compounds with many hydroxyl groups make up the category known as carbohydrates. It may also comprise their counterpart, which upon hydrolysis result in such compound. They are also known as saccharides, and they are most prevalent organic molecules in nature. Sugars are carbohydrates that are dissolved in water and have a sweet flavour. Carbohydrates supply energy to your body, which is one of their key purposes. Before entering the bloodstream, the majority of the carbohydrates in your diet are processed and turned into glucose. The glucose in your body is absorbed by your cells and used to make a fuel substance called adenosine triphosphate via a series of complex processes known as cellular respiration. Cells can then employ ATP to power several metabolic functions. Formation and breakdown of polymers. Polymers are always formed by the polymerization process, in which their basic constitution, known as monomers, react together to produce polymer chains, i.e. 3-dimensional networks that form the polymer links. The nature of polymerization process employed is determined by the biomolecules groups connected to the reactant Almost all macromolecules in biology are either entirely polymeric or composed of long polymeric chains.
Lipids are fatty, waxy, or oily. They are hydrophobic, meaning they are insoluble in polar solvents like water and soluble in nonpolar solvents. Fatty acids and glycerol molecules are the monomers used to build triglycerides or phospholipids. A fatty acid is a long chain made up of hydrocarbons which consist of carbon and hydrogen. A glycerol molecule is made up of 3 carbon atoms and 3 hydroxyl groups which contain oxygen and hydrogen. 3 fatty chains will attach to a glycerol molecule to create triglycerides, as we can see in this picture, or 2 fatty chains and a phosphate group will be bonded through a glycerol molecule to create a phospholipid, as in this picture. Lipids act as hormones, transcription factors, fuel, support for structure, and surfactants. In triglyceride, the long chains of carbon result in more storage for energy, and the breakage of bonds will release more energy. In phospholipids, the positioning of the polar and nonpolar molecules allow phospholipids to protect the inside of cells and act as a barrier between cells and the aqueous environment. Here we have a bilayer with the phospholipid heads on the outside, which tend to be polar, and the tails on the inside, which are nonpolar. When attaching, a condensation reaction takes place and an ester bond is formed between the glycerol and fatty acid. In the formation of triglycerides or phospholipids, 3 water molecules are normally released. This means that when water is added, a hydrolysis reaction takes place and the triglyceride is broken into a glycerol molecule and 3 fatty acid tails. We will now discuss all things enzymes, including structure, function, external factors that affect enzyme activity, and some theories regarding enzyme and substrate activity.
Structure and function of enzymes. At body temperature, enzymes operate as catalysts to catalyze biological reactions. They are known as biocatalysts because they can speed up various responses in the body. Living cells create enzymes to accelerate biological reactions both inside and outside the cells. Enzymes are also known as biocatalysts. Substrates are the reactants in an enzyme catalyzed reaction, while products are compounds generated by the reaction. Activation energy. The lowest amount of extra energy required by a reactive molecule to be turned into a product is described as activation energy. It also defined as the smallest amount of energy required to activate or energize molecules or atoms in order for them to conduct a chemical reaction or change. With adequate energy, molecules can be mixed or converted into many products.
In 1894, Dr. Emil Fischer proposed the lock and key model theory. The analogy is as follows. Imagine the substrate is the key and the active site of the enzyme as the hole of the lock. The key needs to be specifically shaped to fit into the hole of the lock. In terms of biochemistry, the substrate is shaped to fit the enzyme that will be breaking and forming the end product. In 1958, Dr. Daniel Koshland challenged this theory with the induced fit theory. He argued that although active sites are still shaped specifically for a substrate, the active site will still change shape until it is completely bound to the substrate, and hence only the substrate truly determines the enzyme-substrate complex.
External factors on enzyme activity. The rate of reactions between enzymes and the substrate catalyzation can be affected by many conditions, the big 3 being temperature, pH, and concentration substrates.
Temperature increase results in more enzyme and substrate collisions. This increases the chance of catalyzation. Enzyme reaction rates are typically at its peak from the range of 37 to 40 degrees. Enzymes have an optimal temperature of 40 in the human body. Approaching optimal temperature, activity decreases and eventually stops. This is because enzymes will lose their shape. This is known as denaturation, where bonds between proteins within the enzyme are broken.
pH. Enzymes also have an optimal pH in which reaction rates are at their peak. Enzymes in humans have an optimum pH of 4 to 8. Slight changes in pH balance on either side of the range may slow down reactions, whereas extreme ones can denature enzymes. pH refers to the potential of hydrogen. Within an aqueous solution, hydrogen ions also bond with R groups within amino acids. As amino acids can make up enzymes and specifically active sites, if enzymes are not within their optimum pH range, this can interfere with how they bond with substrate bonds.
Substrate concentration. Higher concentration of substrate result in a higher chance of enzyme and substrate collisions. Enzyme reaction rates reach a maximum when substrate concentrations are too high. This leaves enzymes fully saturated. As you can see in the graph, the reaction rates begin to plateau.